Hello, everyone. As we have everybody filing into the room here, I'm going to turn off the waiting room so people don't get stuck in limbo trying to join the meeting. Thank you all for joining me and Dr. Copland, or as his friends call him, Al. And we're here today to discuss some treatments and updates in kidney cancer research. So Al, Dr. Copland, thank you so much for joining me today. We're real excited to have you on the call.
Thank you, Scott. So I'm Al Copland. I'm a scientist at Mayo Clinic in Jacksonville, Florida, and I'm delighted to talk to you and ask questions when you need to, and I'm going to ask you a few as well. So Scott has been told to unmute when I ask questions, and I expect some answers from everybody. So as I said, I'm a scientist here in the Cancer Biology Department at Mayo Clinic, and my task today is to talk to you about basic science and how it can drive discoveries and with a focus on treatment for kidney cancer. So what I'd like to do is tell you a little bit about basic concepts that can help you understand cancer better. Also about the kidney cancer subtypes and cell of origin.
Just up the salmon, Debbie.
And histology and genetic mutations and how these genetic mutations can dictate and guide therapy. And the kidney cancer genome is druggable, but one therapy does not fit all, as you'll see. And I'll give you some examples of basic discoveries leading to life-saving therapies in kidney cancer, and also talk about the future and how discoveries can lead to cures. So getting to understanding basics of cancer, what you have to understand is that we're made of cells, and we all start off as one single cell. And about 100 billion cells later, or 9 months later, we end up as a human being. And so cancer essentially is a disease of cells malfunctioning, and they continue to replicate. And I would imagine all of you guys understand that intimately, but it's a very important concept to understand that our cells replicate, and cancer cells can replicate as fast as 16 hours to 24 hours. So if you think about it, if a cell replicates once every 24 hours, in 30 days, you can have 1 billion cells. Now we're made up of 100 billion cells. So you can imagine what happens with a single cell metastasizing to the liver or the brain and replicating and essentially destroying the function of your organs, and death results from that process of metastatic relocation and cells replicating in your body. This is a beautiful picture showing cell replication, and here essentially are the different phases of mitosis or cell replication, from interphase where DNA replicates. So you're seeing DNA in green, and a cell starting to replicate into two parts. You can see chromosomes forming here, and in metaphase, you can see the chromosomes are aligning with the red spindle down the middle. And in anaphase, you can see the chromosomes separating, and then you can see two nuclei forming into two brand new cells as a single cell replicates itself into two cells. So that's essentially what cancer is doing.
No, I don't think so.
So,
No, no thanks. No.
Scott, you may want to mute everybody. So we're made up of 4 chromosomes, or essentially 6 billion DNA pairs in each one of our cells. And so this is where mistakes are made within our DNA. We have mutations within DNA that causes cancer. And as you can see here in the X and Y chromosome, you can see that the Y chromosome, which men have, there's quite a bit of DNA missing. So you can understand why women are smarter than men. So in our cells are made up of cytoplasm and nucleus, and within the nucleus is where our chromosomes and DNA reside. And within the nucleus, as you can see, are the 46 chromosomes, which are made up of DNA, and that DNA is what different strands of those DNAs become mutated and lead to cancer. And so different strands of DNA are made up of genes, and we're made up of about 22,000 genes that are transcribed into messenger RNA. And it transcribes into 150,000 or more messenger RNAs. These messenger RNAs are then translated into proteins, and it's the proteins that make up the structural components within our cells. And so you can see how there are thousands and thousands of ways or mutations that can occur within any one cell, and you know that we're made up of about 100 billion cells. So it's a miracle every day that we function fine and are able to live fairly long lives. But you can see how the statistics for mutations to occur that become that can turn into cancer are very high. One concept that's important to tell you about is this idea of do we inherit genes, cancer genes? And inherited gene is a mutated gene that you would get from a parent, and that gene would be mutated in every one of your cells. That's a very rare event and occurs in less than 5% of cancers, and typically it will show up in pediatric cases, and so it's a fairly rare event. So most cancers are what we call sporadic gene mutations. That is, it's caused by environmental factors and how we interact with the environment. So many cancers are actually preventable, and so it's how we interact with our environment with regards to smoking or our diet, our lifestyle, whether we're sedentary or very active. And you can see here that there tobacco and our lifestyle can really dictate whether we get cancer or not. And so the American Cancer Society estimates actually that there are 42% of newly diagnosed cancers in the U.S., which is about 750,000 cases, and about 42% of those cases are avoidable. And so they have identified that about 19% of cancers are caused by smoking, and about 18% are caused by combinations of body weight, alcohol consumption, and poor nutrition and physical activity. So again, how we live our lives can dictate our susceptibility to cancer and whether the environment will impact our genetic makeup and specific cells. And so this little yellow dot indicates a mutation in normal cells, and that mutation again has a growth advantage, and so it can grow and cells can replicate. And you can see the yellow cells replicate, another mutation occurs, and as we build up mutations, we build up we go from normal cells towards precancer cells to cancer cells to metastatic cancer, which is the most aggressive cell that has the ability to replicate, survive, and metastasize to distant organs. So about 95% of all cancer deaths are due to metastasis. So if you're diagnosed with breast cancer, you're not going to die from the breast cancer that's growing in your breast, but you're going to die when it metastasizes to your brain or liver and takes over those organs and shuts them down. So we have this process in our body called homeostasis. That is, cells are born and cells die. And so, so about every second, we have about 25,000 cells that divide in our body. But again, we have 25,000 cells that die as well through a process called programmed cell death. And so cancer cells forget how to die, and they continue to replicate, and they're essentially immortalized and can live forever. So as I tell some folks, you know, if you know that you've got 25,000 cells dividing every second, so some days that you feel like you wake up and you feel like a new person, you just might be. So there really are hallmarks of cancer. That is, there are genes that when they're regulated incorrectly, that is, they're overexpressed or suppressed, and combinations of genes can lead to cancer, lead to cells that can replicate and live forever. And so these are some of the hallmarks of cancer. That is, cells have the ability to continue to proliferate or replicate, which I showed you. They have the ability to evade growth suppression. They have the ability to invade and metastasize to other organs. They continually replicate. That is, they're immortal. They can live forever. They induce angiogenesis, so all of our organs need oxygen and blood and nutrients. And so cancer cells have the ability to induce blood vessels so that they can get nutrients from the blood. And they also have mechanisms to really resist cell death. So when we treat with drugs, very often they have mechanisms that can resist cell death from drugs. One mechanism is that drugs can get transported back out of the cells with these multi-drug resistant transporters, that the drug gets in and it gets picked up and gets taken right back out of the cancer cell. So those hallmarks are actually regulated by very complicated or sophisticated signaling pathways. Those 22,000 genes that I told you about make about 150,000 proteins, and these proteins essentially work together in signaling pathways to regulate cell replication, cell survival. And so here you can see that these are proteins in this pathway that regulate cell motility. And so E-cadherin, beta-catenin, TCF4, and you've got other genes and proteins that regulate differentiation and viability. And so these complicated or sophisticated signaling pathways made up of proteins essentially are targets for therapy. And that's what's so exciting over the past 10 years, is that we have made dramatic discoveries of these proteins that play a role in cancer biology and developing therapies against it. And so you can see RAS and MYC are critical proteins. They're called oncogenes because they're amplified and they promote tumor growth. And we're currently working on therapies that target these two big critical oncogenes. And we expect to have therapies against these two targets probably in the next 2 to 3 years in clinical trials. And so, as I said, we can target these hallmarks of cancer. We now have EGF receptors, therapies that target EGF receptors. We have therapies that target glycolysis. We have PARP inhibitors now that are showing survival benefit in patients with ovarian cancer and breast cancer. You know very well, we have VEGF inhibitors that prolong survival in patients diagnosed with kidney cancer. We have c-MET inhibitors, another set of inhibitors that are important in papillary kidney cancer. And so you can see that we are we are fairly rapidly now, because of the technologies we have, developing therapies against molecular targets that are providing survival benefit to patients. So with regards to kidney cancer develops from normal renal epithelial cells. And so genetic mutations occur, we get localized disease in the kidney. Currently, surgery will cure your disease if that kidney is taken out or the tumor is killed. You can also kill the tumor with heat ablation or cryoablation. And I told you that additional genetic mutations occur that drive a localized tumor in the kidney to become a metastatic tumor, to progress to a metastatic tumor. So we still don't completely understand all of the switches that drive carcinogenesis and progression for kidney cancer. And importantly, we also don't understand the links between the risks. There are three major risk factors for kidney cancer: obesity, high blood pressure, and smoking. And we still don't genetically understand the links between that and development of kidney cancer. And so these are areas of intense research. This is, again, another example of signaling pathways. This dark line is a cell membrane, and you can see on the cell membrane we have proteins that sit and cross the cell membrane. These are signaling pathways that allow signals to be generated from outside of the cell into the cell that promote cell proliferation. Apoptosis is a programmed cell death, DNA repair, survival of cells. So the Egf receptor, there's a ligand called epidermal growth factor, or EGF, that binds to the Egf receptor that stimulates cell proliferation in cells. And as I said, the Egf receptor can be amplified or mutated where it is signaling all the time, and it is a very important therapeutic target in a number of different cancers, including kidney cancer. And so again, we're developing therapies that target important signaling pathways on the cell membrane as well as within the cell. Again, this is a diagram on the left of kidney cancer, and normal cells within the kidney and within the proximal tubules and distal tubules are the sites of origination of the different subtypes of kidney cancer. And you can see on the right a papillary kidney, multiple tumors of papillary kidney cancer in this specific kidney. These are histological subtypes of kidney cancer. Clear cell, of course, is the most common, about 75%, with papillary 10 to 15%, followed by chromophobe, and then benign oncocytomas. And so the histology tells you a lot about the cancer itself, and that they truly do have different genetic profiles, which will dictate different therapies for these different histological tumors. And at the bottom, I'm showing you the origination of the cell type, and each of these cell types express different sets of genes, and those different sets of expressed genes will dictate therapy, because those genes that are being expressed can be overexpressed or suppressed. So every cell in your body has a subset of those 22,000 plus genes that are expressed, and gives each cell its unique signature of its function. In this particular slide, I'll share my slides with you so that you don't have to read all of this junk, but essentially these are mutations that commonly occur in the different subtypes of cancer. So with clear cell renal cell carcinoma, the most common form, the 3P arm of chromosomes are frequently lost. And so these mutations dictate the type of cancer that you're going to get, and also our ability to develop therapies that target these different mutations. So what I've said so far, and why is what I've said so far important? And I've said essentially genetic mutations and gene expression alterations are therapeutic targets, and our cell of origin and genetic mutations that are altered, that also have altered gene expression, direct us towards potential effective therapies. And so what I'm proposing to you is that knowing the biology and the genetics of our own cancer will direct therapies that can lead towards cures. So here's an example of papillary kidney cancer, and there are two types, a type 1 and a type 2. And in the type 1 form of papillary kidney cancer, c-Met mutations are quite common. And so c-Met is a ligand for the Egf receptor on the cell membrane and drives cell proliferation. And so you can see that c-Met is located on the 7q arm of the number 7 chromosome, and its mutation can drive and be part of the cause of papillary, this subtype of papillary renal cancer. And so we now have therapies that target c-Met. And on the right-hand side of your screen is a list of a number of drugs that target c-Met, and some you will recognize are also FDA approved for kidney cancer now, like cabozantinib that targets c-Met, but it also targets VEGF, RET, KIT, AXL, TIE2, and FLT3. And so these are now FDA approved drugs, and some are in clinical trials that prolong life and have a survival benefit in patients. And to the left, you can see that HGF is a ligand that binds to the MET receptor that sits on the cell membrane. It then signals to multiple signaling pathways, from AKT to JNK to FACS signaling to MAP kinase. And all of these signaling pathways regulate cell survival, cell transformation, motility, and cell cycle progression or proliferation. So you can see that a single pathway or a single mutation in this c-Met receptor can drive a cancer phenotype, because it regulates many signaling pathways downstream of its signaling pathway. So what I'd like to do now is tell you about some fundamental discoveries that have led to new therapies. And this one happens to have won a Nobel Prize back in 2019 with Bill Kaelin, Sir Peter Ratcliffe, and Greg Semenza. And what did they discover? Well, they discovered an oxygen sensing pathway. And kidney cancer is, and clear cell renal cell carcinoma is the poster child for this signaling pathway. And what we know, and what most of you probably know, is that von Hippel-Lindau gene is mutated in about 80% of clear cell RCCs. And it regulates the expression of HIF-1 alpha, or hypoxia, HIF-1 alpha and HIF-2 alpha in their protein expression. And so VHL actually, collaborating with oxygen, hydroxylate HIF-1 alpha and HIF-2 alpha and drive the protein to degradation. And so in times of hypoxia, that is lack of oxygen, HIF-1 alpha and HIF-2 alpha are allowed, or proteins that are no longer degraded because oxygen is not present. And these transcriptional factors regulate about 200 or 300 genes that promote cell survival. Okay, so these studies started in the early 1990s with Greg Semenza identifying and cloning HIF-1 alpha. Bill Kaelin was studying von Hippel-Lindau tumor suppressor gene, and he cloned this gene, and then Sir Radcliffe demonstrated in 1999 the association of VHL with HIF-1 alpha. So you can see how these basic discoveries in biology can be critical to cancer, potentially to cancer therapy. And so here are a number of genes that the HIF-1 alpha and 2 alpha regulate that promote angiogenesis or blood vessels, drug resistance, this MDR1 gene, erythropoiesis, genetic instability, glucose metabolism, immortalization. So you can see that if HIFs are dysregulated, that they could be bad news in the kidney. And as I told you, von Hippel-Lindau gene, or VHL, is mutated in about 80 to 85% of clear cell RCC. And so there have been a number of drugs that directly inhibit HIFs. And here you can see mTOR inhibitors, HDAC inhibitors, topoisomerase inhibitors, microtubule stabilizers, and HSP. But they're not as effective as a new therapy that's recently been developed at the University at Texas Southwestern in Dallas that specifically targets HIF-2 alpha. And you can see this is a diagram of the protein of HIF-2 alpha, and you can see this small molecule PT2385 has the ability to bind specifically within this three-dimensional pocket of HIF-2 alpha, and it blocks the ability of that protein to act. And so it loses its biological activity and its ability to regulate and transcribe genes. And so here you can see in February 14, 2020, there's a big announcement. Clinical trial exposes deadly kidney cancer's Achilles heel. This HIF-2 alpha protein that was discovered at UT Southwestern is now in clinical trials. And you can see in these photos that these little red dots, which are indicative of blood vessels and angiogenesis, are inhibited by this HIF-2 alpha antagonist, PT 2385. And so you can really see where basic fundamental biology discoveries have led to discoveries of novel compounds like this PT 2385, and finally make it to drug and clinical trials. And you can see this started back in the early 1990s, and in 2020, about 30 years later, we now have therapies, but it's all grounded in basic biology and understanding this basic biology. And so all of the discoveries and therapies that we now have available to us in kidney cancer are a product of these basic discoveries. And so you can see the tyrosine kinase inhibitors that were discovered became FDA approved between 2005 and 2016 are all targeting signaling pathways that are critical to cancer biology. So targeting VEGF and c-MET, the mTOR pathway. And we now have the immune checkpoint inhibitors that target PD-1 and PD-L1 to short circuit immune suppression that's caused by tumor cells. So tumor cells make this protein called PD-L1, and it blocks the ability of CD8 positive PD-1 cells to initiate contact and destroy cancer cells that are expressed that it can target through a pro-tumorigenic immune responses. It looks like I'm running out of time here, Scott. So should I wrap it up? So again, these molecular targeted therapies that we have available to us now in kidney cancer are targeting molecular targets that are critical to the survival of kidney cancer cells. And so we clearly need new therapies, and essentially I'm going to skip a bunch of stuff because we're out of time, and just skip, because we've got a new molecule that we're developing targeting fatty acid metabolism. That's a signaling pathway that's critical. So I'll just skip to the summary slide. So we're moving, very rapidly towards precision medicine. Each one of us with cancer have a unique genomic profile of genes that are mutated or altered that are driving our cancer. And so the key thing is to understand which of those genes are driving the cancer. Do we have therapies that can target those genes? And the idea is that we can use combinatorial therapies that target different pathways to shut these signaling pathways down and don't allow the cancer cells to live. And so, as I said, we've got the technologies now that we can really begin testing combination therapies and develop new therapies as well against a number of molecular targets that we do know that are oncogenic. So we are on, I really do feel that we are on the verge of cures based on this strategy of developing therapeutics that target very key signaling pathways and proteins and genes that are essential to survival of cancer cells. And so you might envision one day either taking a blood sample, because we now have technologies where we can measure cell-free DNA in the blood. So this is DNA that's come from your tumor cells, and identify mutations that are in your tumors, and using the knowledge of the DNAs, the genes that are mutated in your tumor, and knowing and having therapies that can target those mutated genes or amplified genes, we can put together combinations of therapies that potentially will prolong life or even potentially lead to cures. And so this idea of designer individualized cancer therapy, and of course in combination with immune therapies that are coming on, that are on board, as well as other new ones, there really is, over the next 5 to 10 years, there are going to be some incredible discoveries and therapies that are going to come to the clinic to save lives. And so, we're all here for one big reason, and that's to better understand our cancer and to beat it. And so we're on the road to do that. And I want to thank each one of you for listening today, and I really consider each one of you in this group, that is, you're the few caring people that dare to change the world. And this is one of my favorite quotes from Margaret Mead. So I'll stop there and take any questions that we might have.
Thank you so much, Al. At this point, we're open for questions. Everyone should be able to unmute themselves if you've been muted previously, just because I muted everybody because there was some background noise. So you should be able to unmute yourself if you want to ask a question that way, or feel free to type into the chat box. Oh, I see Deb.
Hey, can you see me?
I did for a second.
Okay, I'm not quite sure what I did, but I'll try this again. Hey.
Hey.
Hi, Deb.
Hello, Dr. Copland. I had a question, and maybe you answered it in the last slide after I typed it, but my question was around the percentage of kidney cancers specifically that you think are probably hereditary. So I know the slide said less than 5% of all cancers. That's when you were speaking in general terms, but for kidney cancers, what you think that percentage is? And then just to go on from that, for patients who are going on now, I think there's a lot of buzz that everybody should have genomic sequencing done, learn more about your tumors, but then in reality, everybody's just getting the same treatment anyway. So pretty much people, you know, if you're first line, you're gonna get Pembro-Axi or Ipi-Nivo. So even if you've paid to have that genomic profiling, nobody's really giving you a targeted recipe based upon that. That's my observation anyway. Is that fair?
That is fair, Deb. So the first question is with regards to familial inheritance, and it is true in kidney cancer, it's about 5%. You've got VHL, you've got c-Met mutations that are hereditary, and you've also and there are other sporadic mutations that occur. On the slide that I've got up right now, in young people, you've got this TFE3 gene that can fuse, and it's not hereditary, but it's sporadic, but it can really affect young people and preferentially males, and it's a gene that a number of us are studying now for pediatric cancers. With regards to your second question related to doing genomic profiling, and I think the point that I'm trying to make here in this talk too is that rapid progress is being made, and that very often patients are buying time with the current therapy they're on, and that they're going to rotate through therapies as they progress or become resistant to one therapy or another. But knowing your genomic profile is going to put you, more in the driver's chair when new therapies come available, and you'll be more, you can move more rapidly with understanding or being able to potentially get in clinical trials as they come up with these new therapies. It's like this new HIF2 alpha inhibitor, 2 years ago or a year ago, that was not available. It is now. And we know that HIF1, HIF2 alpha is a driver, oncogenic driver in clear cell RCC. The same way with c-Met. So knowing, inhibitors of the c-Met pathway were not available 3 or 4 or 5 years ago, but they are now. And I think that's the point that I'm trying to make, is we've got the technologies now, and a lot of new discoveries are in the pipeline. So we do genomic profiling of patient tumor tissues and discovered SCD1, steroid-CoA desaturase 1, is a gene that's overexpressed, highly overexpressed in kidney cancer, and there aren't any therapies for that. And so here you can see SCD1 is, the brown staining is overexpressed in every stage of RCC, and we made this discovery, over 10 years ago, and have mapped its signaling pathway, and we've now developed novel small molecule inhibitors specific for SCD1, and are trying to get these into clinical trials. So you're very right that it's not, knowing your genomic profile is not going to benefit you immediately, but being aware of it and trying to learn and understand how these genes work, and potentially there will be clinical trials potentially in the near future that might target key genes that have been mutated in your specific cancer. That was a little long-winded. I apologize for that.
could I just ask a follow-up, Scott?
Yes,
Would I be able
ma'am.
to ask a quick follow-up?
Yes, ma'am.
So the question for a lot of patients is when to do this, because if a patient is going to pay out of pocket, say for Foundation One, or I'm in Canada, so we can do this usually under a clinical trial and get the profiling done.
Right.
And it may not be helpful now, but part of the thinking is that the actual sequencing is getting better and better over time. So when do you have this done? Let's say if you were diagnosed with a small renal mass, that doesn't seem to make much sense. But
No.
do you wait until you're out of treatment options and then say, oh crap, I should do some sequencing,
Yes, because you, as you say, you cannot move any faster than drugs are FDA approved out of clinical trials, and so unless you've got an oncologist who's really thinks outside of the box and is being proactive about sequencing and looking for alternative treatment strategies, I mean, that would be the time to do it, like you said, when you're out of options. Because, there are other issues, there are potential tissue heterogeneity, there's tumor progression as you can develop new mutations based on drugs you've been on. So you're right.
It sounds like, though, getting something like that done could act sort of like a key to helping researchers understand exactly how a treatment is affecting someone across the board if you were to get that genome done. Is that right?
Yes, and many many scientists and physicians are sequencing tumors and linking genetic mutations to therapeutic responses, and also looking, as I said, looking at tumor heterogeneity and trying to understand drug resistance and development of drug resistance and identifying different signaling pathways that could be later targeted or important to the disease.
Mm-hmm.
That
sounds like the largest jigsaw puzzle I've ever heard of.
It is. I mean, that's why we haven't cured cancer. I mean, it's not a single gene disease. Especially solid tumors are, you've got a litany of genes that are mutated. So we did an experiment a number of years ago. I don't know if you still got the slides up that you can see, but we
can see them.
Yeah, so we did genomic profiling and just took the top 200 genes that were overexpressed commonly in clear cell RCC. And we have technologies that we can silence any gene that we want in cells. And so we can do this in cell culture in the laboratory. And so we silenced each one of these top 200 genes. Just the top 200 genes, we identified 31 genes that promote cell proliferation in kidney cancer cell lines. 31. So knock one of them out, you've got 30 others that can promote. And granted, many of these genes happen to be in the same signaling pathway, which is great, so that targeting part of the one of the pathways can be redundant. But again, you've got multiple pathways that are redundant that are driving any one cancer. So it's a pretty complicated, sophisticated disease for solid cancers.
Mm-hmm.
Thank you for your question, Deb. Does anyone else have any questions for Al? James Smith had said Opdivo and Yervoy, but I'm not really sure what the question was there.
It really wasn't a question, as that was the treatment that I was on for almost 2 years, and that had it was successful in basically preventing my tumors from enlarging. Though I did have some tumors grow and then disappear in my lungs over time, but I did have a new round of tumors starting to grow in my sac. I have bone metastasis, sorry, to my spine. So I did have a new tumor growing there, and it's pressing on the nerves coming out of the L5 S1 area. It's causing me to have a drop foot. I'm having surgery for this tomorrow to go in and actually just clear out that particular area, take pressure off the nerves so I can, hopefully walk right. But yeah, my oncologist is moving me towards putting me on a clinical trial, which includes the Cabometyx and a drug called PT 2977, I believe. I don't know if that sounds familiar.
Say that again, James. I didn't hear it.
PT 2977.
Right, that's the new HIF2 alpha inhibitor. Right.
Okay. That's what he's going to be putting me on. Once I kicked my Kepler habit, because I had a brain tumor, which caused oh Lord, the terms are escaping me right now.
Yeah.
Seizures caused me seizures.
Right.
So, but yeah. But for a while there on the Yervoy and Opdivo, I was doing real well.
Right.
But then my cancer, I guess, got smarter.
Or, as I said, your cancer can be heterogeneous. That is, it has parts of it have a set of mutations, and another tumor cells have a different set of mutations, and so your immune checkpoint inhibitor therapy may have been able to take some out, but not all of it out. So that's another possibility as well. So, but I would love to. I'm going to share my slides with Scott, who can send it out. And my email is at the bottom of the last slide. So I would love to hear how you do, James, and keep up with you, because I'd like to really see how this new HIF2 alpha inhibitor is doing.
Okay.
So,
in addition to that too, I did have a brain tumor which caused the seizures. I did have a gamma knife procedure which obliterated it, and there's been no trace of it since on MRI. I've had radiation to my lumbar area, and that seems that it had just stabilized the cancer, which is okay. But I had the last round of radiation last October, and it took away the pain that I was having then, but it doesn't look like it did anything else, because I have new growth basically in the same area, it seems.
right.
So we're pretty sure that my cancer doesn't care about radiation. So
Yes.
that's why we're going with the new clinical trial.
Yep.
Thank you for sharing that with us. We all of us, I believe, would be very interested to see how your surgery goes or your procedure goes tomorrow. And so if you'd like, we'd love to follow up with you and check in and see how things are going.
Oh, that'd be fine, yeah.
If you could shoot me an email, or you can send me a message on the group chat here directly to me with your email address. I'd love to exchange information.
Okay.
Don Bain had a question. How long until we can expect these therapies to be in practitioners' hands?
If the therapies are in clinical trials, it could be 1 to 2 or 3 years, depending on accrual and performance within a clinical trial. As I said, we've got a small molecule inhibitor targeting steryl CoA desaturase, and we're at a point where we're doing all of the toxicology studies that have to be done to get FDA approval for investigation of a new drug. So this IND approval by the FDA is absolutely necessary for any compound to go into a human being. And so the FDA is mostly interested in whether it is safe to go into a human being. So there are a lot of tests that we have to do to make sure that there's a predictable safety profile before a compound a human can take a compound. And a drug is not a drug until it has been administered to a human. So so it may take us, another year to get through the FDA approval if everything works as planned. And then you're at a stage, and that typically takes anywhere from $2 to $4 or $5 million to get there. So funding is a big issue. Once you get to clinical trials, you've got to pay for a clinical trial as well. So this is a homegrown discovery. So we discovered it here in my laboratory. And so we're footing, all of the bills through federal grants that we've written, and we're partnering with a small biotech company to help commercialize our compound. And so clinical trials can take, 2 to 4 or 5 years, again, depending on how they accrue and do in clinical trials, before they can show, if they show survival benefit, then you submit your data to the FDA and request the FDA for FDA approval of that drug for specific treatment of patients of the indications that you've been doing in your clinical trials. So it all takes time and money.
Yep, and the more people that we have participating in these clinical trials, the more quickly this can happen.
Yes, and I can tell you, clinical trials are much better designed today. They've got better rationale with regards to preclinical data for therapeutic strategy, and so I think we're, drugs that are getting into clinical trials have a better chance of success and benefit to patients, because we understand so much more about the human genome and cancer biology and signaling pathways, and so it's not like the old days where you had a drug and you just tried it because you had it. We've got so many more options now, and tests that we can do and understand that a drug most likely will have benefit in a patient.
If you're interested in learning more about these, please make sure that you reach out to your primary care physician and ask them about clinical trials. You can also, there's some great information available online at clinicaltrials.gov. And you can reach out directly to some of these hubs like Mayo Clinic and University of Miami Sylvester Center, and these other places around the country and all over the world actually that are holding these clinical trials. There's a wealth of information available online, and our support group online would be happy to help you finding something in your area. You can reach out to us on our website or on Facebook. I'd like to take a moment to recognize that tomorrow is World Kidney Cancer Day. So I know that IKCC has been putting together a campaign talking about how important it is to focus on your health and wellness and being physically active, as that can play a primary role in boosting your success rate in battling kidney cancer. And our foundation has put together some stories and thoughts and feelings of other patients that people have shared with us. So we'll be posting that tomorrow if you guys are interested. I will also post this video on our website and our Facebook page. And Al, or Dr. Copland, has offered to share these slides with us. So those will be posted tomorrow as well. Let's see, have any more questions? Okay. Well, if anyone does think of anything else, you can always reach us online. So thank you all for attending. Next week, we'll have another round of our kidney cancer conversations with Dr. Roberto Peeley. He will be sharing information on the importance of diet in a kidney cancer patient's life. So please tune in for that. And if you have any suggestions on topics that you would like us to cover in the future, we would very much like to continue this series on a monthly or twice a month basis. So please feel free to reach out. Thank you all for attending, and I look forward to seeing you soon. Thanks again, Al. You were great, and thanks for answering everyone's questions.
Thank you, Scott. And like I said, feel free to email me if you have any questions. I'm happy to answer anything I can. Or if I don't know it, I'll find the answer for you.
Excellent.
Oh, one other thing someone had asked is, how do we register for Dr. Peeley's talk? You can do that on our site. If you go to jnfkidneycancer.org, on the homepage, you should see a link to register for that event. All right, everybody. Thank you so much and have a great day.
Thanks, Scott. Bye-bye.
Bye, Dr. Copland.
Mm-hmm.